to start to look at the nucleus of the cell, and we're going to look at how cells divide. Right? And so we're going to kind of combine some different processes that are taking place in the cell and focus a little bit on one, then the other, and then kind of go back and forth. So the idea here that we're really starting off with, to put it in perspective, is cell division. So all cells come from other cells. Cells divide, and then they give rise to new cells, and then they give rise to new cells, and they give rise to new cells, and so on. All right, so that process of cell division is going to then give us, two new cells, and then those cells will divide, into new cells, and so on. Typically, a cell divides into two, but there are cases, as another type of cell division that we'll talk about later, which is called meiosis, where we actually end up with four cells. There's sort of two divisions happening, one directly after the other, and we get less chromosomes in those cells. This is to make the gametes in particular. So we only really see that type of cell division in a certain type of tissue. So not all tissues and not all cells are capable of meiosis. Only germline cells, which means the cells that make sperm and egg. And so only in organisms that reproduce sexually do we even see meiosis. So some organisms that reproduce asexually do not undergo meiosis. Prokaryotes, they do not undergo meiosis. A number of different organisms do not undergo meiosis. But mitosis is common for pretty much all cells of all living things. And that's kind of what we're going to be focused on. So the first part of this that we're going to get into is, what has to happen in order for the cell to divide. Well, inside the nucleus of the cell, we have our DNA. And so right now I'm just going to draw one little squiggle representing the DNA, representing the chromosome, which different cells have many different numbers of chromosomes. Our cells have 23 different chromosomes, but we have 2 of each, so we have a total of 46 of them. So before this cell, though, can divide into 2 new cells, though, let's say it just has this one chromosome, just as an extreme example. If it were just to divide in half, that means one cell would get the chromosome, the other wouldn't, and this other one without the chromosome wouldn't even survive. So before there can be any cell division, you have to double the DNA. All right. So this one chromosome has to double, right? And we have to double the DNA before we can have this division process. Once that division process occurs, then we can get a new chromosome into each of these cells. If another division is going to take place, then we have to double the DNA again, double the DNA, and then we can divide these equally into other cells, and so on. This process would have to keep occurring and occurring and occurring over and over and over again. The thing that we're going to pull out of it right now to focus on is where we double the DNA, and this is a process called DNA replication. And I introduced it a little bit earlier on when we talked about DNA structure, but now we're going to start to get into a little more detail. So the first step to doing this is to look at the overall process, sort of a general overview of it, and then we're going to start to get into detail of the enzymes that are involved in the process, their specific jobs in that particular process. So you'll need to know the names of the enzymes, the jobs of all the enzymes, at a certain level of detail. There's a lot more detail that we're not going to get into here. This is still considered sort of an early level information on replication. It's not an advanced course, but we need to know these sorts of things. So DNA is what we're going to start off with. The chromosome. So some terms, again, this goes back, it's just a little bit of review. So a chromosome is our genetic unit. Right. An entire chromosome is going to be double-stranded DNA. All right, and cells can have multiple chromosomes. That means they have several separate double-stranded DNA molecules. In addition to the double-stranded DNA, there are proteins, organizational proteins called histone proteins, that are also part of it. And a chromosome is really about 50% DNA, 50% protein by mass. So that's a review. go back if you're not familiar with the specifics of the structure. Go back and look at the earlier lectures on DNA. So what we're going to go through now is saying DNA is being double-stranded. Okay, so this is the strand, has nucleotides. And again, you should review that material. So the nucleotide, remember, it has a 5-carbon sugar. I'm trying, it's kind of small because we have other stuff to draw on. This is just a review here. There are, say, 1, 2 in this ring, 3, 4 carbons in the ring. The 5th carbon is outside the ring. The carbons are labeled with a little prime, which I'll talk about again in a little bit. On the number 5 carbon, the 5' carbon, has a phosphate group. The number 3, or the 3' one, always has an OH group. And to the number 1 is where we have the nitrogenous base. And that nitrogenous base is what's unique about each of the nucleotides. And that nitrogenous base can either be A, T, G, or C in DNA. And in RNA, there's the U instead. But we're looking at DNA right now, DNA replication. So this particular strand of DNA sticking off of one end would be a phosphate. If we had multiple nucleotides, at the other end would be an OH. Okay, we would call this our 5' end, and this would be our 3' end. Now, assuming the structure, we're just going to simplify it. Instead of redrawing the sugar, every time, we'll just have the sugars are in here. But sticking off the sugars, which are exactly the same and repeating, phosphate, sugar, phosphate, sugar, phosphate, sugar, what's unique about them are the nitrogenous bases. So we might have an A, a T, a C, another C, a T, a G, an A again, and so on, thousands and thousands long, right, of these nucleotides. Now it's a single strand, but DNA is double-stranded, double-stranded DNA. So the other strand of the DNA here is going to be, if you remember, antiparallel. So antiparallel means the orientation of the phosphate and 3' OH is opposite. So at this particular end, up here, we're going to have the OH. That's the 3' end. And down here, we're going to have the phosphate, the 5'. So phosphate, 5', OH, 3', 3, 5. So they're opposite one another. They're what we call antiparallel. Now, if we look at this strand over here, what we're going to have is T, A, G, G again, A, C, T. There are base pairing rules, which hopefully also you remember. Adenine forms 2 hydrogen bonds with thymine. So A and T pair up, and G and C form 3 hydrogen bonds. So these are hydrogen bonds. Always 2 for A and T, 3 for G and C. So 2 here, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2. So this is our teeny little segment, but double-stranded DNA, showing us, reminding us of some of the important parts of the structure, right? That there are 2 strands, that the strands have 3', 5' ends, that the 2 strands are anti-parallel compared to one another, that the bases pair up, and that the bases pair up with hydrogen bonds, and there are specific rules in which they pair up. A and T bind, G and C bind. Okay, so now that we have that structure, we start to get into the idea of replication. So in DNA replication, it's what we call semi-conservative replication. Semi-conservative. So that means we're going to conserve whole strands of DNA. They're not going to be broken up into bits or pieces. We're going to have whole strands. But this original DNA, if this is our original parent strand, once it's broken apart, it's never going to come back together again. Okay, so it's going to stay separate. So it's not completely conservative. This double-stranded DNA is going to be broken apart and not come back. But the strands themselves will stay together. So what's going to happen here, so I'm going to now erase one of these strands, is we're going to say that something comes in, and we're going to get to what that something is in a the next part of this, as we're going to add into the detail, something's going to come in and break the hydrogen bonds between the strands. And just add it in right now anyway, we'll go into it. That thing that comes in, it's an enzyme. It's an enzyme called helicase. Okay, so helicase is going to be the enzyme that comes in and actually breaks these two strands apart. Like I said, we'll get to the detail of that a little bit later. And so I'm redrawing a strand that's been broken away, pulled away from the other side. So with no hydrogen bonds now between the two strands. If they were allowed to just sit there, after breaking the bonds, they would just come back together again all on their own. Okay? The hydrogen bonds are attractions. They're polar attractions between partial positive, partial negative parts of those nucleotides. And so they would just reattract and join each other. Again, as we add in detail, there's going to be something that we refer to as an SSBP. SSBP is going to stand for a single strand, SS, binding protein. So it's going to be a protein that binds to single strands of DNA and keeps them single strands so they don't come back together again. So that's going to be part of this process too. But now just kind of getting into the overview of it. Double-stranded DNA is pulled apart into two single strands. Each single strand in semi-conservative replication is a template. So now this is a template. This is going to be a template. What does that mean? That means that there's going to be an enzyme, which we're going to, again, talk about the details of it later on, that's going to work along this particular DNA. Now what it's going to do is it's going to start to build new DNA as it works. And it's going to start to add one at a time new nucleotides here. See, one at a time, another G, an A, the T. It's going to add these nucleotides in. And how is it going to know which nucleotide to add? Because it's just the base pair rules. If there's an A here, it has to bring in a T. If there's a G over there at the template, it has to bring in a C, and so on. So there's only these particular options. The way I drew it, I happen to draw it like this, from 5' to 3', and that's the way that the replication process is going to work. The same thing is going to happen for the other strand. So along this strand here, all right, we're going to get a nucleotide with a phosphate. Then it's going to have an adenine nucleotide come in. Say a thymine nucleotide come in. Cytosine, that should be a C. 3 bonds. Another cytosine, 3 bonds. Thymine, 2 bonds. Guanine, 3 bonds. Adenine, 2 bonds. OH. Okay. So if you look at these, then side by side, and compare them, AT, TA, CG, CG, TA, GC, AT. Now you have 2 double-stranded DNAs. That's the overview. That's our semi-conservative replication. We need that so the cell can divide. So before the cell can divide into 2 new cells, we have to double the DNA. We started off with 1 double-stranded DNA. Now we have 2 double-stranded DNAs that are exactly the same as one another, hopefully. Errors in the replication process will lead to mutation. So you've heard of mutation. There are a number of things that can lead to mutation. Some that can artificially accelerate the rate of mutation, but mutation's a natural process. And that's essentially where a mutation is where you would say have T, like that. It's a mistake. It means that in the DNA replication process, actually I should do this the other way around here, where this was the G. I'll put a T here. So this was the original, the G, and then the new strand, the one that we built by accident, the enzyme who did this put a T in here. Now that T and G don't bind. Right? That's a mistake. It may be fixed or it may not be fixed. If it's left like this, that means part of the genetic code in the new DNA has been altered. Right? It's been messed up. That is called a point mutation. And it may have a major effect on that particular cell's future, or it may have very little to no effect on the cell's future. It just depends on exactly where it happened. But that would be a mutation, would be an error in the copying of the DNA. So it's misreading the template and then putting the wrong nucleotide in there. So that's the overview. That's the basics of DNA replication, semi-conservative replication, and I've mentioned a few of these enzymes. So what we're going to do now is we're going to actually get into the real details of the process. Looking at DNA replication, where we actually go with the enzyme step by step, one by one, and then look at when they come in, what their specific jobs are, and how DNA is replicated. That'll be the step 2 of DNA replication. And then step 3, we're going to have another lecture after that that goes through some additional details, some troubleshooting, things that happen afterward to process the DNA before mitosis can occur. And then we'll actually get into mitosis after that. Okay, so that's kind of the overview of what's to come. So the first part here is just more of a reminder of nucleotides, DNA structure, base pairs, and how the overview of replication will occur. Then we're going to get into the details next.